Tier 2 in Arizona University. He has also had high school course, Oceanográficas from isolate to genomes, where he teach high school students how to obtain in 2 weeks what took him 2 years after his PhD. And also he organized the environmental virology workshop, cutting-edge experimental informatics and theoretical thinking, attended by 50 participants from 8 countries. So let's receive the virus guy who works with viruses, marine viruses in the middle of the desert.
Thank you very much, Consuela, and thank you, Osvaldo and Kurt, for this great opportunity not only to speak today, but to be involved in this great course. It's been an excellent experience for me. I clearly love this stuff. It's been great to get back in the lab and work with students to isolate viruses. I want to share with you today what I feel like is maybe what Woes went through about 30, 40 years ago. I'm going to try to describe to you some biology that we've learned, and I'm going to do that by telling you about the methods we had to invent to learn this biology. And this is to see what viruses are doing in nature, and particularly focused on ocean viruses. So we've heard a lot already about Woes as a revolutionary and a visionary, and he was able to put together some challenging data sets of very complicated nature and run those through complicated computational algorithms and come up with some sort of organization for life. And this is really a paradigm for us to work from for probably decades, and it's a great organizational scheme for us to be thinking about how to organize life. But I would posit that Woes really wanted to study viruses. If you look at his thesis, his PhD was on the biophysics of inactivating viruses, and actually some of his early papers were on, of course, his PhD work, but he also was interested in the virus-host interaction. Here he was studying bacterial viruses, or phages, and how they interacted with a bacterium he was interested in, Bacillus. Well, so why didn't Woes study viruses? I would posit that's because there weren't tools, he couldn't see them, and they probably weren't important. And before 1989, that was true. If you went to the oceans, tried to isolate viruses, you got nothing. So of course, there's not many viruses. And this was because people used cultures to isolate viruses that unfortunately, we now know, are not really abundant hosts in the ocean. So if you take E. coli to the middle of the ocean, you're probably not going to get a virus. But if you take Prochlorococcus or Synechococcus to the middle of the ocean, you might get viruses. We were dead wrong. There's not no viruses out there. They're very abundant. We heard about the astronomical numbers for microbes already. 10 to the 29th microbes in the oceans. There's probably 10 to the 30th viruses in the oceans. There's about 1 million to 100 million viruses per milliliter of seawater. If you add them end to end, you end up getting 10 to the 8th light years away from the planet. There's more mass in viruses than there are in all the cows on the United States or all of humanity times 10. There's about 10 viruses per cell. I used to put prokaryote, quickly changed that. So 10 to 1 is a number to keep in your head, 10 viruses per cell. And this picture here actually is seawater concentrated on a filter that's been stained with a DNA stain called cyber green. And hopefully what you can see is big dots, and those are the cells. And little dots, those are the viruses. And hopefully you say, yeah, there's about 10 little dots per one big dot. Viruses are not just abundant, they're also very dynamic. They turn over on the scale of every few days if you do tracer experiments. Woes had written a piece towards the end of his career, this is in 2007, with Nigel Goldenfield, I think reflecting on his early biological revolution experiences with microbes, and now sort of being excited about all the gene flow or horizontal gene transfer being observed in genomes. And I was a graduate student at the time and was just ecstatic when I realized he cited 4 papers, and one of them was a paper that I'd just published. And the reason he wanted to include that in this story is because viruses have a big role in moving genes between cells. In this case, we'd found that viruses that infect photosynthetic cells, cyanobacteria, actually encode the gene right at the core of photosystem. So this is the photosystem is where water is split to make hydrogen and oxygen. 4 Nobel Prizes have been won studying this little PSBA protein, and we found the PSBA gene in our viruses. And if we looked at the evolutionary history, and I could see none of my animation is working. If we looked at the evolutionary history of these viruses, what we found is that you have one group of viruses up here. These are Synechococcus viruses. Another group of viruses, Prochlorococcus myoviruses 1 and 2, and Prochlorococcus podoviruses. And this PSBA gene sequence in this tree from viruses clustered with their hosts, suggesting the virus stole that gene from the host. What was interesting is that this happened 4 times, 1, 2, 3, 4. And if we looked at whether there was any shuffling of that gene or intragenic recombination, we could see that viruses were sharing the version of this gene a lot. They could share it across viral types, even across myoviruses and podoviruses. And viruses could actually take the gene from the host and use it to their own end and put it back in the host. And this matters when we step back to think about this host, because this is a cyanobacterial cell. All it really wants to do is float around in the ocean and fix carbon dioxide into sugars using light energy from the sun through a process called photosynthesis. But the virus has come in and injected its genome. Part of its genome is that core photosynthesis reaction protein. So the virus has shut down that cell's ability to do anything, and it's boosted the cell's photosynthetic machinery, not really to make that cell continue photosynthesizing for the cell's sake. The virus is doing this just so that the virus is able to make a lot more progeny. And we've shown that through a series of 10 other papers that the virus has stolen this gene and it's selected for in the virus because it helps make more virus. But 30% of the cells in the environment are infected by a virus at any given time. That means 30% of the cells are under a photosynthetic regime which is totally different than the rest of the cells. So this impacts global biogeochemistry, in particular through carbon fixation and oxygen here. And what I'm not telling you is that we've discovered a lot of other host genes related to phosphate stress, nitrogen stress, sulfur cycling in viruses. And so viruses have a way of interacting with their hosts, but they also, in this new revolution that Woes was referring to, do it in a very subtle way by manipulating core metabolisms to their own end, but at the same time altering that cell's output. So this is the outline of my talk. I will introduce the role of ocean viruses in 3 slides. And then I'm going to try to tell you what I would consider to be the new viral revolution. We're developing an ability to see and to count. That may seem very simple, but boy, am I glad we're finally able to do that. Okay, so to introduce viruses, again, they were thought to be non-existent in the oceans, and then 1989 we looked at them on a grid and found there's probably 10 to the 31 globally. So with all these abundant viruses, what could they be doing? In the 1990s, there was a lot of effort to try to figure that out. And so the first thing we think of is that viruses kill cells. They kill about 20 to 40% of bacteria per day, or about 3 to 10% of cyanobacteria per day. And then this is another big number. They move about 10 to the 29 genes globally per day. So viruses pick up a gene and can move it from one host to another 10 to the 29th times per day on the globe. If you look at a microbial genome, about 3% of that genome is actually viral DNA. And if you look at the community microbial genome, about 3% of that DNA is actually viral. And this ranges from 0 to 18%, depending on which particular microbe you're in. Not only are the genes there, but they're often some of the most highly expressed genes. This is just showing us a genome, a microbial genome. Black genes are microbial genes, and the red is to schematically highlight a prophage, an integrated virus. And the red shows that those viral genes are highly expressed. And great examples of that are the toxin genes. Cholera toxin, pertussis toxin, Shiga toxin, Bordetella toxin, many toxins we associate with microbes. It's actually the virus in the genome which encodes the toxin gene. And when the virus's regulatory capacity gets out of whack, that's when you have a toxic outbreak. If we look at this from the oceanographic perspective and look at carbon flow in the ocean, you've got the oceans are a net carbon sink at about 3 gigatons per year, bringing more carbon dioxide in than they let out. That cycles through the larger cell loop, so larger phytoplankton, which can feed larger animals called zooplankton and fisheries, or smaller phytoplankton, which can feed both the bacterial loop and the microzooplankton. And you can have organic carbon, which sinks out of the water column or comes down to the deep system through diel vertical migrators. And we were just beginning to discover the microbial loop in the early 80s and mid-70s, as Carlos nicely outlined earlier. And the viral shunt was just posited in the late 90s. And so generally it's thought that viruses keep carbon small. So viruses lyse cells. That organic material becomes available to smaller cells rather than moving up this food chain. And I'll just flag that we desperately need measurements. Biogeochemical measurements of the impact of viruses are really from the mid-1990s. There's very little work done on how viruses manipulate fluxes in the oceans. And then I'll just remind you again that viruses impact photosynthetic evolution and photosynthetic capacity through that photosynthesis phage story I told you earlier. So what's this revolution? Well, I'd like to be able to see more than dots, because if you actually start to think about how a system works, 100 dots or 10 dots per cell doesn't get you very far for making any predictions. If those cells behave in different ways, if those viruses behave in different ways, it doesn't get you very far. So the first thing we did was simply develop a quantitative electron microscopy method to be able to turn those dots into pictures. We used this to test a hypothesis about what kind of viruses are out there. So the hypothesis was that tailed viruses are the majority in the oceans. And so here we've got tailed viruses. We've got a classic myovirus, which is a long contractile tail. A cyphovirus, which is a long non-contractile but flexible tail. And a podovirus, which has a tiny little extensible tail. And if you look in culture collections, 96% of these viruses of bacteria are called phage. 96% of these isolated phages are tailed. So the expectation was that the ocean should be dominated by tailed viruses. So we tested that hypothesis with this Tara Oceans sampling, where we took samples from 14 different stations, 6 oceans, and looked at 2,600 viruses. And we found that only 1/5 of all the ocean viruses are actually tailed. So we completely reject this hypothesis. Most of the field is studying tailed viruses, and yet there are only about 20% of the viruses that are out there. The second way to be able to see is to start to link viruses to their host cells. So this is a method called phagefish, and it targets single copy genes to be able to link viruses and hosts as follows. Essentially, you have a fluorescent in situ hybridization probe, fish, probe to the host cell, to 16S, the ribosomal RNA we've heard a lot about, and another probe to a phage gene. And then what you can do is look for co-localization of the red and green dots, and that lets you know that that cell is infected by that virus. And that method is great because it allows you to simultaneously count the number of viruses that are out there that infect a particular lineage, and you don't need genetics. A big problem in doing this kind of work in the oceans is that there are no genetic systems for most of these host cells. So this is completely, this is accessible to almost any cell that you can normally use fish with. So I think this means these green dots turn into many colored dots, which might belong to viruses of different lineages. You can imagine dots related to SAR-11 viruses and SAR-116 viruses, Prochlorococcus viruses, Synechococcus viruses, Bacteroidetes viruses, Vibrio viruses. A lot of the common host cells you can imagine being out there. And this enables a lineage-specific free viral ecology. You can look at the viral particles in seawater and start to get a handle on which viruses infect which hosts. A third way we're learning how to see is simply to ask, how do we organize unknown sequence space, or viral dark matter? What is unknown sequence space? If you go out into the environment and you sequence the viruses, 90% of the data are going to be orange here, which means they match nothing in the databases. So these are from 4 sites, the Sargasso Sea, the Gulf of Mexico, British Columbia, and the Arctic Ocean. You can see that about 90% of the data we can't say anything about. Well, this was in 2006. We redid this in 2013 with better technology, better library prep methods. Unfortunately, the databases aren't any better. We're still completely in the discovery phase. 90% of what we see when we sequence a virome is new to science. Well, this is exciting to me because we can organize that sequence space. This is what your metagenome, your fragmented viral sequence data from the environment might look like, just an unorganized mess. And then you can organize that into bins. The bins here we call protein clusters. And this allows us to map about 70% of the reads from any given sample into a protein cluster. And once you've got something in a protein cluster, it turns out we actually increased the global map of viral sequence space twofold just in our first analyses. And this allows us to have a unit for diversity, which I'll show you a slide on in a second. It allows the field to start to functionally annotate things. So let's say a protein cluster were really abundant in many places around the world, and it turns out this is true. There's 180 protein clusters in all viral communities that we've examined so far in the Pacific Ocean, and 2/3 of those have no known function. So they're abundant and shared by every Pacific Ocean virome, yet we have no idea what they do. Those are great targets for synthetic biologists and biochemists, geneticists. Also, you can do ecology of protein clusters, and I'll talk about that shortly. I just want to highlight that all of the source code to generate protein clusters, all the protein cluster data themselves are currently at CAMRA. When you can't actually download the data from CAMRA, you can contact me, and I will make sure you can get it. And by May 2014, these data and the pipelines to generate your own protein clusters will all be available at cyber infrastructure located in Arizona called iPlant. We've adapted iPlant for iVirus and iMicrobes so that we can have informatic pipelines available globally. So in light of sort of these big global numbers and how many species are out there, I thought I'd just throw in our own effort on the viral side. This is a last-minute addition. In 2003, Forrest Rower suggested there were 2 billion viral proteins globally, and this suggests, this is in every paper pretty much that's written, it says viruses are the largest genetic reservoir on the planet. And I'll just tell you that if we revise the data, now that we have more than 14 genomes, and look at this with a fresh analysis, this is with a bunch of metagenomic data, it looks like we were probably only off by 3 orders of magnitude. There's probably only 4 million viral proteins globally. These are the data. So we've got 32 Pacific Ocean viral metagenomes. That's part of a data set called the Pacific Ocean Virome. If you do these collector curves that we've seen a couple times, the rarefaction curves, it just shows that we actually saw 456,000 protein clusters, and Chow estimates there could be something between 456,000 and 1.3 million protein clusters. There's all kinds of assumptions made in the paper, challenging extrapolations, and the implications, and they're described in this paper, which is now available. It's highly speculative, but we've since actually gone back with Tara Ocean's data, and we're definitely flattening this curve now. In green are the protein clusters that are novel. So this is the pan genome that Norm spoke of earlier. And so now we're beyond estimations and we're into observations, and I'd say we're pretty close to saturating that curve. And that's for all the Tara Ocean's viromes, which surface to depth, plus all of our in-house data. We only see about 1.1 million proteins. And the extrapolation is that we think about 1/3 of the diversity is in the oceans and 2/3 in the planet. So you get to something on the order of 4 million proteins globally. So the fourth way to see is simply to be able to develop new model systems. So this is a bacterial, a tree focusing mainly on the bacterial phyla. And in red are the things in culture, and in black are the things that we have a sequence for, but no isolates in culture. And then we've stacked on here where the viral isolates come from. And what I hope you can see is it's very clear we're biased towards just a couple of phyla. 85% of 1,100 sequenced phages, bacterial viruses, are derived from only 3 out of the 45, at this time, 45 known bacterial phyla. So what happens if you actually start to look into those viruses of other bacteria? Well, this was an effort we did in collaboration with Steve Giovagnoni. His group led this work. He's the one who can culture SAR11 that was brought up earlier. So this is the first look at SAR11 viruses, and they're called pelagiphages. And it turns out that if you use a new host that's abundant, you get the most abundant viruses known. So HTVC are the high throughput viral collection viruses, and this is a potovirus from his collection. It's a SAR11 potovirus, and I just highlight the other potoviruses. My cyanophage, which had been the most abundant, went from first to second with this paper. And then another paper came out, the SAR116 phages, also another abundant host group from one of Steve Giovagnoni's postdocs, came out in PNAS this year, and it's the second most abundant. So I went from first to third in the span of just a couple months. That's okay. And then my postdoc, in light of Carlos's talk earlier, my postdoc Karin Holmfeld, who's now got her own position in Sweden, had isolated Bacteroidetes phages and found that they're representatives of what we call the rare virosphere. So she's seeing viruses which are ubiquitous. We can find them almost any virome, but they are not very abundant anywhere. I'm sure we'll find conditions where they're abundant eventually. So I think just in this year alone, we've gone from very naive to the most abundant and second most abundance now being represented, and a representative of the rare virosphere. So again, this is going to be very exciting. We're going to learn how to see already, and now we're going to learn how to count. So the first thing you could count are genes. If we count genes, we're going to call that gene ecology. And the problem was that most viruses are not culturable, just like most microbes. And the culture-independent surveys we'd used to survey in nature were not quantitative. Basically, they either were too small scale in the case of the linker-amplified shotgun libraries. They suffered from cloning bias and from being just a couple thousand clones. Or they were Phi29 amplified. And DNA that's been Phi29 amplified has a stochastic bias, meaning a non-reproducible bias, that usually skews single-cell genomics projects 1,000 to 10,000 fold. So I like to be a lot more accurate than 1,000 to 10,000 fold. And then for viruses, Phi29 also has a systematic bias because it over-represents small circular DNA. And so it's going to, and single-stranded DNA. So it's going to really change the viral community when you look at it. So we spent about 4 years developing what we call a quantitative sample-to-sequence pipeline, where we just tried to better improve how to concentrate viruses. Instead of using tangential flow filtration on the 200 or 400 liters, we now use iron chloride chemistry-based chemical flocculation. The community used 3 different purification techniques, and we evaluated all 3 of those to figure out which ones were the right ones to use. And instead of using 529 amplification, you can use a linker amplification method. And these are all published in the 3 primary papers. And then Melissa Duhaime wrote a review with me to just describe that whole pipeline. And Sergei Solenenko has worked on, this is now published as well in BMC Genomics, a comparison of all the different sequencing platforms and what the resulting biological output is. So we understand very well now how to create a quantitative virome, and we're plus minus 1.5 fold. So you still could be off as much as 150%, but that's much better than plus or minus 1,000 to 10,000 fold. Okay, so what could you do with this Pacific Ocean virome? We've got samples from the entire globe now between Tara Oceans and Malaspina, and we're focusing just on the Pacific Ocean. Have you ever heard that before? Focusing just on the Pacific Ocean. And when we look at these data, Bonnie Hurwitz was a student who did this work. We found some really incredible results. These Pacific Ocean viruses seem to nearly completely reprogram all of central carbon metabolism. So if we look across diverse pelagic ocean waters, viruses have genes everywhere there's a colored arrow. And so you'll notice that wherever there's shortcuts in central carbon metabolism, viruses often take the shortcut, particularly stress-related shortcuts. So just like the photosynthesis story, viruses of probably many other different hosts are tying into the key metabolism that limits viral production during infection. So to summarize our ability to count, we've got a summary of this gene ecology section is that we have quantitative metagenomics now. We can now count these different genes because we've got an organizational tool called protein clusters. And that's already showing us that viruses broadly manipulate microbial metabolisms. And the inference, though it would be great to get some measurements, is that this must impact ocean biogeochemistry in a very big way. So the second way that we could count is by looking at organisms. And I'll just warn you that we can't just go out and PCR amplify a gene. Not one gene is shared by all the viruses. And so you could start again just by counting dots, which I suggest gives you minimal information. You could turn those dots into characters in the play, the poto, myo, sipho, and non-tailed. And I'll just let you know that in that same paper I showed earlier, Jennifer Brum also elucidated ecological drivers of viral community structure from that global sampling. And it turns out temperature and salinity are major drivers. But if we really want to get at predictive power, I think we need to get at virus-host interactions, and we need to understand the genome, the underlying genome of the virus, because that can tell us so much about these metabolic influences. The problem is that there's this paradigm that viruses exchange genes so rapidly that it erodes any phylogenetic lineage. And so this is a plot from a paper from Roger Hendricks in 1999 with some viruses here in the different corners and linking them by some of the genes that are thought to be shared. And so you can think of this as just this web of gene flow between different viruses. And we've seen this before in the microbial world. Ford Doolittle, for about a decade or so, has really pushed that horizontal gene flow between even domains could erode taxonomic inferences. And I think in the microbial realm, I can confidently say that there's consensus that there are lineages there and there's good taxonomic information, so I had hope. And specifically, I had hope in answering this question. If we go out and survey in nature whether what the variability in genome sequence space looks like for viruses, is it continuous or discrete? In other words, if we could plot genome sequence space, is it just one big continuous blob, or are there discrete populations? And this is important because we want to be able to count something. I can't really count something here because what would be the different things? It's also, and so that's for ecology. It's also important that they be a formally defined population because we want to be able to think about the work in an evolutionary context. So I've been interested in a long time in the simple question of, I have a single host cell, how many viruses are infecting it? I can count maybe 4 viruses here, but I'd like to know what are those different types or not. So we answer that question by going into the environment with a single host and asking, how do viral genomes vary in nature? And is variability continuous or discrete? Again, because we hope to count something, and count something that's evolutionarily meaningful. So we knew a little bit before we started this project. I had isolated 2 viruses from the same seawater sample on the same host. Don't like to extrapolate very much from an N of 2 for the oceans. But what we'd learned is that these 2 co-isolated viruses shared about 4 out of 5 genes, and did so, each of those genes that they shared were about 83% identical at the amino acid level. Well, then we decided to do the experiment again. 4 new Synechococcus cyanophages, and they only shared 1/3 of their genes, and at much lower identity. Okay, this tells us there's a complexity we don't understand yet. And we're never going to get there with cultures. Isolating hundreds and thousands of viruses and sequencing their genomes is way too much money, and any graduate students would kill me. So this is a method we call viral tagging that Phil Hugenholtz actually gave me the idea for and worked with us on through this project. It started in 2008. And the concept is simply we're going to experimentally label wild viruses with a cybergreen stain and just ask them to label their hosts for us. And we're going to try to pull wild viruses out of the environment with a bait host. So if you do this, you mix wild viruses with your clonal cellular host. You get, without the viruses, you get just cells. These are cyanobacteria, so they autofluoresce red. And these are photomicrographs from the Amnis image stream. So each of these red dots is a single particle triggered on a flow cytometer. And then if you add viruses, you get red for the cyanobacteria plus green, which are the labeled viruses. And if you're doing this on flow cytometry, that means that you can actually have a green shifted population. These are the untagged cells, and these are the viral tagged cells. And then you can sort out the viral tagged cells, do a trick to get rid of the cellular DNA, and keep the viral DNA. Sequence that, and you've got population genomes from the wild. And this took a long time to ground truth it, but now it's working particularly for cyanobacteria. So what did we do with that? We went out to an oceanographic transect called Line 67, starting from Monterey. It's a beautiful nutrient gradient. And we just asked what viruses infect this Synechococcus host using viral metagenomics to screen about 10 to the 7th viruses. Anyone who's ever tried to isolate viruses, you don't want to isolate that many. That's a lot of work. So we did 10 to the 7th in 10 minutes. And this is the population genome landscape plot. If you've been a little sleepy because it's after lunch, try to sit up. This is a neat set of data. This genome landscape plot just plots an arbitrary sequence space, one axis against another, using principal components as the underlying statistic. And I've first plotted 4 genomes that are from 1, 2, 3, and 4 that are from isolates. So we actually isolated these cultures from the seawater, and you can see their spread. And then we add in genomes that we got out of, or genome fragments out of the viral tagged metagenomic data, and they're the orange dots. And the clouds around the data are the variability in the underlying population. So resampling the metagenomic data to get a sense for how big this population variability is. What I hope you can see is that in no cases do these clouds overlap except right here and partially right here. And on a third axis, which explains another 8% of the data, those are well separated. So what we're resolving here are many different populations that are clearly distinct, discrete in this genome sequence space. Interestingly, all the work from my thesis is right here in the gray. 10 years of PhD and postdoc work from isolates is a tiny fraction of that genome sequence space compared to this one 10-minute experiment. So what did we learn? These cyanobacterial viruses form discrete groupings in the wild. That suggests there's population structure that we could count. In this case, there's 20 viral populations at least that infect this particular host. Each of these genome fragments has underlying metagenomic sequence data. So you can count and get counts for each of these populations. So you can quantify these populations. That's ecology. And it's a population-based ecology. Here we learned, although there's a lot of issues with it, that the local diversity, the viral tagged data set, is much larger than the globally sampled isolate diversity, because this is from many hosts and many seawater samples in gray. And interestingly, the cultures, 1, 2, 3, and 4, actually represented the 1st, 9th, and 10th most abundant populations of 26. So I looked at this and said, you know, cultures represented the most abundant, but they also represented some of the lesser abundant cultures. So if you only have one culture, you may have done really well, and it may be representative, or maybe you've done less well. But I was very comforted by the fact that we got the most abundant in culture. This also tells us something about a species definition. If you look within these populations, you can look at variation again in the metagenomic data. And I hope you can see this axis. It's only a few percent away from 100. So if you have a metagenome and you've got a sequence read which maps to a reference, it better be within a percent or two, or else it's not really from that population. We can see as much as 4% variation in the more established populations, or less than 0.5% variation at any given locus in the perhaps more recently clonal bursts. So in this plot, you've got reads that people in the field would typically say, oh, these are T4 phage, and they probably are, but they're definitely not this population. Only the red metagenomic reads that are being recruited in this fragment recruitment plot are related to this population. So the summary here is that viral genome sequence space is clustered. We see discrete populations. We've developed an operational viral species definition by letting nature tell us where those breaks are, and that is that the average nucleotide identity should be about 98% or higher at each locus. And we've got an estimate of between population variability, as well as numbers for each population. So we can do population-based ecology, and we went back now to this transect at a few depths and a few sites, again using our host in this viral tagging method. And you don't need to know anything here except that these y-axes represent abundance, and each one of these panels is a different genomotype, or a different viral type we pulled out of that seawater. And hopefully what you can see is that across these 6 sites along the transect, different viral genotypes have different abundance profile distributions. But what's neat is that each of these, again, represents a genome. So now I can say, oh, this one's really abundant. I wonder what's different in its gene set. What might allow this thing to exist? What might drive its ecology? And so what's interesting is as we started to mine these data, because it's pretty new, these genomes suggest there is ecological differentiation, and you can see that by the different abundance patterns. And it's driven by phosphorus, nitrogen, and of course, many unknown genes in these viruses. So these are great synthetic biology targets. These are driving population distributions, and many times the abundant populations across this transect. So come back to Carl Woese and just say that maybe if he were born today, he would have studied viruses. I like to think that he's excited about this totally unexplored world in the same way he probably was when he first started launching into an understanding of the biological organization that we're all currently using. I want to summarize in just this one slide and say, I hope you've learned that viruses are abundant, about 10-fold more abundant than the number of cells on the planet in almost anywhere you look. I hope you see that I just highlighted a few ways in which they play critical genetic roles by manipulating things as fundamental as core photosynthesis, as well as manipulating ocean biogeochemistry in ways that range from lysing cells to altering a cellular infection status to altering the actual photosynthetic biochemistry through gene conversion. And I hope I've inspired you that there are many new ways to see through informatics, through the microscope, through new culture collections emerging, and a suite of tools that should allow you to begin to think about doing viral ecology to complement any microbial science you want to do. And we can do that in a lineage-specific manner and linked back to your host that you care about. And just some names of some of those methods. I didn't talk about targeted viral metagenomics, but I mentioned phage fish, quantitative transmission electron microscopy, quantitative metagenomics, viral tagging, protein clusters, etc. This has been the work that I've driven for many years, but it's happened not in a void, but with great microbial collaborators ranging from Debbie Lindell on the photosynthesis work, Stephen Hallam and Alex Warden on 2 great oceanographic transects, Phil Hugenholtz for inspiring and working with us on the viral tagging work, Rudy Amon drove the fish expertise and the phage fish work, Steve Giovagnoni on the pelagiphage, sequencing through a number of consortiums, some proteomic work I didn't talk about. This is a great way to identify the unknown. We identify about 10% of the average unknown sequence space just through proteomics. And then I tried to highlight my lab members whose work I was highlighting as I went along, and just thank funding agencies. Thank you for your time.
Thanks, Matthew, for this interesting lecture. So now it's time for some questions.
Alvaro?
Matt, you mentioned in your talk that you are down to maximum now of what I call 2 million protein animal. But that's only from the double strain or everything.
So the data are only from the double-stranded viruses, and then there are extrapolations we made and laid out in that paper.
So that you will include the single-stranded
And RNA.
Related to this question, when you do different families, which is related, or the definition that you use?
Yep. We spent years back and forth on, the question is related to the protein clusters and how do you define a protein cluster. It's not a biologically meaningful construct. It's a computational arbitrary artifact. And we worked with a lot of things. It's not very sensitive to the percent identity or percent of coverage cutoffs you use, because that tells me that protein sequence space for viruses is also pretty clustered at the gene level. But it's not a biologically meaningful construct. It's simply a tool to organize and count genes that are related by sequence similarity only. I don't talk about homology or evolutionary or functional relatedness. Go ahead.
And I have a second question that could be more philosophical. What do you think is a virus?
Well, it's nucleic acids and protein, sometimes lipids. You mean the life question?
Yeah.
So I return that question. What is your definition of life? Because the philosophical debate rests on the definition, and until I hear your definition, I can't answer the question.
How they appear?
How-
How they
Well, I mean, it's plausible that viruses could have come before cells, because you could imagine nucleic acids and proteins associating before all the other complex cellular machinery. Does that mean that viruses replicated before cells came along? That's hard to think about. But the world was so different, I'm sure that anything could have happened. I have trouble with the deep branching questions. I don't deal well with the early Earth. I leave that to the French philosophers. Sorry, Danielle. Where are we going?
Now that we know that viruses are not just moving photosynthetic genes, but also other genes related with global carbon cycle and all that stuff, do you think that we can, and it's again a kind of philosophical question, do you think that we can now start to think about that like a viral metabolism?
Yeah, I do for sure. It's not so philosophical. So The question is, can we translate from sort of observing these biogeochemically interesting genes in a virus to something that's a model input, right, to a flux? So we're trying to do that through model systems. So we have the viruses that infect the cyanobacteria in culture. So we're asking simple questions like, how do nitrogen and phosphorus impact outputs, photosynthetic outputs, plus minus virus infection? And you can do that for any of these hosts you get in culture. I think the other way to do it is the targeted viral metagenomic method I didn't talk about, where you can actually target those genes and pull out viruses, particularly for those using some molecular biology tricks. And there it gets a little more complicated because you don't have the host in culture, but you can imagine playing some fun games in silico to at least look at the enzyme chemistry. But for sure, I think there's a lot of room right now for thinking about that virus-host interaction with the specific question of the output of the cell and how it changes plus minus virus infection. Yeah.
I understand that the viral infection depends mainly on the host density. But is there any other variables, environmental variables, like temperature, salinity, or even turbulence that can affect the viral infection?
Yeah, the question is host cell density the only thing that impacts success of the viral infection? No, there's a lot of other things. So in cyanobacteria, they only absorb in the light. So the lights during the day are on, so they'll absorb. And that relates probably to this photosynthetic readiness. We know along salinity gradients, particularly in estuaries, when you go from 0 to 30 or 40 parts per thousand, that viral absorption rates change. And there's probably a couple other ways I'm not thinking of at the moment, but anything related to the host physiology has a chance of impacting viral infection success. There are examples of viruses that get into a cell that is so starved that viral infection will fail, because of course it just can't get enough nitrogen and phosphorus to make nucleic acids and proteins.
There's a continuous improving in nucleic acid staining. Why there's no correlation between trying to use like flow cytometry for detection and sorting and everything in viral particles, trying to use this kind of techniques?
We'll see if Danielle gets too mad at me here, but as soon as you stain viruses in the sea, there's already a background noise, which makes it very difficult to count those populations. And a lot of those populations are populations in the noise of the flow cytometer. Now, there are examples of them being countable and pretty convincing. And recently, there was nice work that went all the way from the sorting to the sequencing to sort of confirm the hypotheses about what these populations are by Willie Wilson's group at Bigelow. And that really, where it's successful, is good for the giant viruses. So the viruses that are bigger have more nucleic acids and tend to have a larger fluorescent shift. So it's just that they're so close to the noise that it's challenging. But in the right hands, on the right flow cytometer, it can happen.
Is the basic microbiology of these SAR11 things known? Is it like a D4 or something like that?
Good question. So these SAR11 viruses, is their basic physiology known? So they look like T4-like and T7-like phages. The myos are T4-like and the podos are T7-like at the level of the genome. One-step growth curves have been done, and they look like any other open ocean, the Prochlorococcus or Synechococcus one-steps. They're very long, not huge bursts. And I would say they're T4-like viruses that have stolen some of the SAR11 machinery to adapt to that host. Azoro?
There have been people trying to put the virus into the tree of life. So what's your opinion?
Do viruses belong in the tree of life? I really just stay away from that question. I almost put it in because Vaulot asked me to be controversial, and I knew he wanted that fourth domain of life in there. You know, I'm going to just keep quiet. I'm happy to talk to you in person about my thoughts on it. I just don't want Danielle to go back and ruin my image in France because I work too much with the Tara Oceans people. Was there another question?
One of the numbers that you mentioned was 10 to 29 transduction events per day.
That's right.
Now, if I combine that with evolutionary processes, could you tell me a case where a metabolic pathway could really be traced back to transduction events?
Could,
transferred from one to another?
are there any cases where the metabolic pathway has been transferred by transduction?
I mean, a simple photosynthesis gene is not photosynthesis.
That's right. Yeah. Yeah.
So is there any likelihood that a process as complex as photosynthesis could ever have been transferred by transduction?
Yeah. No. No. And I didn't. Thank you for letting me have the opportunity to clarify. All of central carbon metabolism is not in one virus. That's definitely, each virus might have one or a couple genes in central carbon metabolism. It infects its favorite host, and those genes that it's got are the bottlenecks to making a lot of viruses. Could I ever imagine viruses mistakenly packaging a whole metabolic pathway and moving it to a new host? I could, not something for the size of photosynthesis, probably, but smaller metabolic, you know, proteorhodopsins and these kinds of functional capabilities.
More piece by piece.
Yeah, it seems improbable to me. I feel like these genes are sticking around in viruses because they're key bottlenecks in making more viruses. So I didn't mention it, but it's not just PSBA in photosynthesis. There's also plastocyanin and ferredoxin. And those are in there because they reduce backup in electron flow away from PS2 and PS1. There's also PSBD, which is the cognate pair of PSBA, and that's in the particular viruses because if a virus infects one host and its PSBA doesn't work with the PSBD, it's going to need its own. You know, so there's a lot of logical reasons for the couple of genes they have. And I guess I don't see major metabolisms being moved around by viruses, but maybe others would think differently.
I think when you look at our genome, you'll see this, but when you look at genetic organization, in some cases, it's clear, it's probable that transfers occur of full pathways. For example, photosystem 1 and proteobacteria. You know, the genes are all linked, and there's 40 or so things that are all in a block. Just like actual fixation, which has been shown to transfer, hasn't been shown to virally transfer. Clearly, it does transfer to cell.
So don't close it out yet. So that's true. Yeah, you could be 40 kb or so. That's about the limit of what I feel like a T4 phage could really handle. And that's your biggest vector for these things. So other than the giant viruses, but in the bacterial realm. Sorry? Well, 49 kb, but that would be a non-functional lambda after that, yeah.
What do you think in the case environment would be other organisms? I am thinking in a symbiotic complex mixture of big organisms inside with a lot of microbiotes from different genera, and all of these could be also infected by virus. What do you think about that?
You're saying as we explore sort of the symbiont in this cell and that cell.
Inside symbiotic associations.
Yeah.
Very broadly.
I think there's no question there could be a role for viruses there, and I would suspect that those kinds of viruses would be the temperate viruses, which can temporarily hang out as a prophage, integrated or not, in the cell, because the host cell abundances would be so highly variable. I think as all these single cell genome projects are coming out, the single cell genome, particularly the larger single cells, end up being metagenomes in and of themselves, viruses being one component. And I think that'll be a real opportunity in the next couple of years to see, to start to get a window into that kind of interaction. for sure it must exist.
where he obtained his PhD in Oceanographics. What to say about Dr. Vaulot? He is an adventure scientist in the field of marine biology, and his major contributions were to understand the ecology and diversity of phytoplankton. His current interests are focused on the molecular diversity of picoplankton in several marine systems and the application of metagenomics to determine the functional adaptation of specific plankton groups. His legacy is beyond the understanding phytoplankton ecology. Dr. Vaulot is one of the major contributors of the Roscoff culture collection, where he continues to supervise. Professor Matthiew, thanks for accepting participating in the symposium and Ecology. We will be happy to learn more from you.
Thank you very much. Thank you very much. Osvaldo said it was my first time. No, it's my fifth time here. So I really am very grateful to Osvaldo initially for inviting me here in Chile for this course, which is really wonderful, and also for Kurt, who is managing it like with a strong iron in an iron hand in a velada glove. Anyway, and I'm really very, very pleased to be here to present you some of our data. And I must say that every time I go to ECODIM, every time is different, and every time is better. I mean, it's really incredible. And where is Matt? I learned a lot about viruses this time. I really enjoy it, and it was great. Okay, so what I'm going to present you, I'm going to be a bit more or less general than most of the speakers here, which draw a big image, either viruses or of the impact of RNA on the field, or the fantastic lecture by Carlos, who is always going, everything is everywhere, and so we. I'm going to focus on one group of eukaryotes that we have been studying for quite a long time now, and I will present you a metagenomic approach to study these eukaryotes. But first, let me set the picture. I like to begin a lot of talks by this picture, which is the surface chlorophyll of the concentration of the ocean. And as you can see, there are some areas where we have a lot of biomass, and then there are some areas in the middle of the ocean where the biomass is the lowest, very low. And in particular, one area where it's very, very low is in the Southeast Pacific Gyre, where we had the chance to go with Osvaldo on a cruise. And I will talk much more about that cruise. In the region where the biomass is large, which are either coastal or upwelling waters, most of the production is due to diatoms or dinoflagellates, large cells, large microbes. But it was discovered that in the area where the biomass is very low, it is dominated by very small cells. And this work dates from the 80s, when it was discovered that it was a mixture of bacteria, of course, of cyanobacteria, Synechococcus and Prochlorococcus, and also of eukaryotes. It turns out that even in coastal waters, this population of very small cells is also present, and it's a background population. Basically, you have this background of photosynthesis made by picoeukaryotes and cyanobacteria, and then you have this bloom of diatoms or dinoflagellates that occur very sporadically. And one technique that we used a lot, so you saw this slide already. Sorry, Carlos, to use my own. But one technique we used a lot is flow cytometry, where you can detect using single-cell fluorescence different population. And if you look at the size of the cells, proxy is size scatter, versus chlorophyll, usually you see three populations in any samples. Any samples in tropical waters, because in other waters Prochlorococcus will disappear. So very small cyanobacteria, Prochlorococcus 0.6 micron, and Synechococcus 1 micron, that Matt spoke about for their virus, because he did his thesis on the virus of Prochlorococcus. And then there is a small eukaryote population. And one of the questions we had, and I'm still trying to answer this question in more details now than initially, was the cyanobacteria. They are very low diversified. At least they belong only to 2 genera. So our question is it the same for eukaryotes? Do we have a single species within these eukaryotes, or do we have a large range, a large diversity, genetic or phylogenetic diversity? So one way we approach this is using cultures. And I really believe, and there I don't really agree with what Norm said, that culture are absolutely critical. We cannot understand an ecosystem if we don't have culture. So we started with culture, but our problem was that we knew that there were a lot of these eukaryotes around, but we could have only very few culture. One of them is Ostrococcus, for example, the smallest known eukaryote, photosynthetic eukaryote, about 0.8 micron. It has everything to make a cell, a nucleus, a chloroplast, a mitochondria, a Golgi apparatus. So it's really the miniature eukaryote. And it turns out that this pico-eukaryote we can find in all the eukaryotic phyla where we have photosynthesis. So we have the green lineage here. We find also, for example, this species, Bolidomonas, which is a close cousin of diatoms, but it's not a diatom. It's within the straminopyle, so it's in the brown algae, so-called brown algae. We found, for example, this species called Florentiella, also in the brown algae. Even in more exotic phyla like Chloarchnophyceae, we find also some very small cells. So there seems to be not in a single branch like cyanobacteria for prokaryotes. Said something wrong for bacteria. You will see. It's going to be. You will see. So we try to understand a little bit better, and for this we had to use culture-independent techniques that the work of Carl Woese has really made possible. One funny thing is that I heard Norm lecture, and then I was looking for a tree, and then I found this in this work, the prokaryotes, where actually this is a paper by Carl Woese, where it's essay on prokaryotic biology, and his name is there. So I don't know what his real message is. Anyway. Sorry? Tough problem is the title of the book. Yeah. But still, it agreed to be the book. Anyway, so let's continue. We apply this approach the first time in the equatorial Pacific. It was about 10 years after Steve Giovannoni has made his landmark paper in Nature, where he had found that a very large fraction of the bacteria corresponded to non-culture bacteria, not non-cultivable, but non-culture bacteria. So we said, well, we are going to try to do the same thing for the eukaryotes. One problem came up. One of the review of this paper, which appeared later in Nature, was, oh, but this is known, you know, all eukaryotes, they can be cultivated because they are supposed to be known and no. And actually, it turned out that what was true for bacteria was also true for the eukaryote in the ocean. A large fraction of them, of the sequence we obtained, some sequence match known species, but most of the sequence, either they were forming different clades compared to known species and very different, or they even could form completely new clades, actually, or even new groups. So what was true for bacteria is also true for eukaryotes. So we are still continuing our our culturing effort, but at the same time we want to understand a bit better what these uncultivated eukaryotes correspond to. So that's why we went to this very interesting area which is the Southeast Pacific, and this is this cruise, Biosop, where we went from Tahiti, Marquesas, Easter Island, and near Concepción in Valparaiso. Here, where you arrived in Concepción, yeah. Yes, the ship arrived here, but some of the samples were taken a little bit. So this is why it's such an interesting area, because you cross incredible gradient from extremely productive coastal waters to mesotrophic waters near the equator to the most oligotrophic part of the ocean, where here, if you put, you use this technique called Secchi disk, you reach the depth of 74 meters. So you are over the board, and 74 meters below, you see your Secchi disk. So it's really a very, very oligotrophic waters. And in this water, I was telling to Matt, we think that Prochlorococcus disappear in the surface. We cannot see by any technique, actually, Prochlorococcus in the surface. So it's very, but still the eukaryotes are still there, actually. So what we did is we used flow cytometry, and we sorted different population, and especially population of pico-eukaryotes. And one postdoc in my lab, Xiao Li, she then did clone libraries. At this time, it was still clone libraries. And to identify which were the population of pico-eukaryotes that we will find in these different waters. And we use both 18S genes and plastid 16S genes from this sorted population, just focusing on the photosynthetic part. So what we found actually is that we were able to establish that the major groups of photosynthetic eukaryotes in this picoplankton were green algae, and especially in the upwelling, we have some Mammilophyceae, so cousin of Osteococcus. And then we have some clades, which are still undescribed, but turns out to be quite important. We call them clade 7. And one is clade 9, is completely uncultivated. There is not a single culture yet. And it's an important eukaryote out there. And then we have brown algae, Stramenopiles. Again, a group, Chrysophyceae, very abundant in fresh water, but is almost unknown in marine waters. There is very, very few species isolated. So the oligotrophic clones or strands are still to be isolated. And finally, another group, Haptophyta, again, many of them, very, very small cells, are still on culture. So like we have 6 major groups, and out of these 6, 3 are still completely uncultivated. And actually, I was, while Matt was doing his talk, I was thinking about the Tara Ocean data. And in Tara Ocean, so covering the whole ocean, we find actually that some of these groups, like Prasinoclide 7, Haptophyte, and Chrysophytes, are extremely important in the picoplankton, looking at the whole global set of Tara data. So what's very interesting with this cruise is like a miniature view of the ocean, actually. So for your next Senate talk, Osvaldo can mention this. So anyway, so we did, 18S, 16S is very good, but as everyone mentioned, you have no idea about what the function of these cells, what they are doing, and we want to isolate them in culture. So that's why we try to do metagenomics, so to know a bit more about the genome. One good thing about these small eukaryotes is that in contrast to diatoms or dinoflagellates, their genome is manageable. The size typically of Osteococcus is about, I think, 16 megabase pair or 17 megabase pair. So we are in the range of 20 megabase pair and not several million like for the dinoflagellates. So we didn't use also single cell sorting like what it's really done now, because it's very, very involved technically, and we do all the sorting on the boat actually. So I don't think this is something you can do on the boat. Single cell sorting, there are, at my knowledge, for marine stuff, only 2 labs in the world that can do it. The one in Bigelow, and now there is one in GGI, I guess, but nobody else can do it. So it's very, very, but still, if you are a little bit more dirty, you can sort several populations actually, but they will be much more, the selection will be much stronger than if you do filtered sample. So we sort cells, we extract DNA, we do whole genome amplification, and then in our case, we still a few years ago, we were going for 454 sequencing. And then we have this pile of data that we don't know what to do. We try to reconstruct things or to make some story out of it. So I'm going to tell you 4 stories, actually. One which is easy, metagenomics. One which is very difficult, and we're still working on it. And 2, a story which occur by serendipity. So meaning serendipity is a mixture of chance, but being also clever. So putting 2 things together and saying, this match. And I think in this metagenomics data, if you go with the big approach, usually it's not so interesting. But if you look for things that are hidden in the haystack, then it can be very interesting. So the first story is about these 2 samples here that we took in the upwelling. So this is work that has been done especially here with Osvaldo and Rodrigo, where we knew from clone libraries that in these samples we had almost pure population of this chlorophyta called Bathycoccus, a small algae which is about 1 micron. 1.5 micron. Bathy, why Bathy? Because it has been isolated from depths. So Bathycoccus, small coccus isolated from depths. So we did half 454 sequence run. So we had about 600,000 reads per sample. We tried to assemble them, so to get contigs. And actually, we were pretty successful at getting long contigs from this data. For example, when you do single cell sequencing, it's very, very difficult to get long contig. Here we were able to get a longer contig. Then we had our contig, and then we did as everyone is doing. So we blast them against the gene bank to know whether there are things in our data that match anything in the database. When we were doing this, actually, it's hard to see here, but most of the sequences are corresponding to bacteria, which were probably associated to the cell, or to noids. And although we knew that in our sample, it was the dominant eukaryote was Bathycoccus, only 10 sequences were matching Bathycoccus. Why is this? Well, it's a bit obvious, actually, thinking about it afterwards. Is that Bathycoccus, the sequence was not available. The genome sequence was available. So although we had it in our sample, even BLAST, although it has 2 cousins, Micromonas and Ostrococcus. And Micromonas and Ostrococcus were in the database. But although the cousins were in the database, we could not find any evidence by BLAST that we could match this sequence. By chance, we have a very good cooperation with people in Banyuls, south of France, who at the same time, so that these people were responsible for the sequencing of Ostrococcus. And at the same time, Hervé Moreau told me, we're sequencing Bathycoccus. I said, this is nice. Can we try to use your data to match our own data? So we worked together. And what happened is that when now we include the Bathycoccus database, almost 30% of our metagenome sequence match something, and match very well, actually, Bathycoccus. So this was really to say that when you have a good reference, and I think we insisted several times on this, then you can interpret your data. And when we knew this, what we did is we went the other way. So we used Bathycoccus as a reference. We had 20 chromosomes of Bathycoccus, and we tried to match our reads, so the metagenomic reads, to the culture Bathycoccus. One point, which will become important later, the Bathycoccus metagenome is from Chile. The Bathycoccus culture is from Metsi, the opposite side of the world. Okay, so very, very large distance. But still, our reads were matching very well. Here what you can see is that you have the longest chromosomes and our two samples. And this is the number of reads which match each position on the chromosome. This is one thing which is very well known with WGA, is that the coverage is not uniform. So you have, when you do whole genome amplification, you have some areas of the genome which are very well amplified and some which are very poorly amplified. And this was the case here, and, but there is not much you can do about it. So anyway, so then we were able to do a very long alignment between our culture, so that's the first sequence here, and the two metagenome, which were from two different samples taken 2 different days in the upwelling. So it's like replicates because it's very mixed water, so but it's not the same sample. It's completely different sample, different CTDs, different processing. And what was amazing, and you can see this here already, is the conservation. And we found that between the culture, strain and the metagenome, at the DNA level, we have from 95 to 96% conservation. So the culture and the metagenome from the ocean are very, very similar, incredibly similar, actually. But what was interesting also that we were able to see also some genetic variability within our metagenome. So here is, again, a consensus for our metagenome. And here it's individual reads. Before we were looking at what's called contigs, so where we put reads together to try to be long sequence. Now we are looking at these 500 base pair pieces that you get from the sequencer. And we were aligning them. And what you can see is that you have 2 types of sequence. This is in one sample, okay? One has a single pair variation with respect to the culture, and one has 3, okay? So in our natural population, we have 2 genotypes of Bathycoccus. Are you following, Carlos? Yeah, okay. So if you are following... yeah. In another sample, so that the other sample a little bit, 1 day later, a little bit further in the upwelling, we were doing the same thing, so our culture reference, and we are mapping the individual read from the metagenome. So these individual reads, you can think them as coming from different cells, basically. So here we had 2 types of cells, with one with 1 mutation and one with 3 mutations in this region. And in the other sample, completely independent, we have exactly the same thing. So we have we have some cells which have 1 mutation and some cells which have 3 mutations. So we can begin almost to do population genetics at the genome level using these samples when you have reference. So, and we publish this in PLOS if you are interested. Okay, so that's the easy part. Not so easy, but it's really interesting. It turns out also that there seems to be 2 types of Bathycoccus. One which is coastal, like our Med-C or upwelling Bathycoccus. And there is another type of Bathycoccus in the open ocean that seems to like the deep chlorophyll maximum. So that's a real Bathycoccus. So the really, well, the other Bathycoccus is a surface. I don't know how you call the surface stuff, but it's not a batty. But anyway, okay, so then we went for the hard samples, which were those either from the gyre or from near the equator, where we have, we know from clone libraries that we have uncultured groups, like this Chrysophyceae, for example, that I told you that we have many species of Chrysophyceae in culture, but they are all from freshwater, virtually none from marine waters. So we run for these. And then we do the same thing. I mean, we do, again, try to match gene bank. And the problem is that it's the same. We find that almost all, like 60% of the sequence have no hits. And some have hits to eukaryotes. But undefined eukaryotes. I mean, it's like they match a little bit each of eukaryotes. So if we use Megan, we cannot really put them in a box, in some eukaryotic box. I'm just going to focus on these two samples. One is from the surface, and one is from the deep chlorophyll maximum. And again, for the other one, same thing. A very large fraction of the reads cannot provide no heat at all. So again, the big problem is the reference genome. By chance, the Moore Foundation has made a project to sequence 750 transcriptome of protists. And among them, there are many of the bugs that are close to our oceanic picoplankton, and some are coming from our Roscoff culture collection. There are some green algae, for example, some straminopiles, some aptophytes, and also some heterotrophs. So what we did is we now compare our metagenomes to these transcriptomes. And what happened when we do this, if we look at, so these are the Batycoccus samples. If we compare the scores that we get with our blast between gene bank and the transcriptomes, we find no improvement because now in the gene bank we have Batycoccus. So we find that we find no improvement. But if we add the transcriptomes, we find that most of the reads we can much better results now because we include things that are not in gene bank that are related to our oceanic bugs. And for example, here, what we find is that 30% of these samples correspond to chrysophytes. Chrysophytes, again, which are not in gene bank, for which we just got the transcriptomes. And this matched really our expectation, because when we were doing tiniest from these samples, we found chrysophytes, actually, uncultured chrysophytes. So this is very, very critical, is to get a good reference to improve. So we are still working on this part. The other part, what we want to do also is functional analysis. So we use a classification for genes called cluster of orthologous gene, COG. I will not go into the detail which one is better because you have several classifications for genes, COG, KEG, and so forth. This is one way. So each of these bars represent categories, and the different bars correspond to our 6 samples that we sequenced, for which we did the metagenome. And it was quite disappointing when we saw that because, so for example, just for the class, here is genes involved in cytoskeleton, here in nuclear structure, and so forth. So you have all the different categories. What was really disappointing is that all the samples look similar. We had some kind of a general profile. So for example, this group here is very abundant. This group is very abundant. So it looks like it was the gene signature for eukaryotes in the ocean, and there was no variation. But then we also looked at the culture for which we had metagenomes, and here is the different culture we looked at. So we looked at the transcriptome. This is a COG analysis for the transcriptomes. And again, the profile looks very similar to what we observed for our metagenomes. But what was interesting here is that we had photosynthetic and also non-photosynthetic, heterotrophic. And we began now to look a little bit, to see a little bit of difference. For example, genes involved in signal transduction are, there are many more COGs in the transcriptomes corresponding to heterotrophic cell, which makes sense because they need like to sense where their prey are and so forth. So we did. So we have now these profiles for both transcriptomes and for a metagenome, and we did what everybody is doing: principal component analysis, or I don't know, one of these multivariates that Rodrigo told us about. And you begin to see patterns, actually, in terms of, so this, the proximity between the different transcriptome and metagenomes is in terms of their gene signature. So things that are close seems to have the same number of genes or same type of genes. So for example, if you look at heterotrophs, they're all grouped together. This is for Peter. This is Eox and Gephyrocapsa, so the coccolithophorids, they all group together. The green algae, they all group together, at least the Mammilophyceae. And then we can see that, for example, our Batycoccus samples group together. They don't group so well, actually, with the Mammilophyceae, but at least they group together. This, which contain green algae, actually kind of cluster with green algae, and these two which contain brown algae also seems to cluster with the brown algae. So we began to see some order, actually, in this metagenome, where initially it was just some random noise. But there's still a lot of work to do, and we can also relate this to the class of gene, actually. For example, here we see that this direction, so the genes that, the samples that cluster here are more similar in terms of energy, genes involved in energy, transduction, nucleotides, and so forth. Anyway, so that's my second story. Where is my timer? I have still some time. Who is timing? Yeah? Yeah, okay. You are my timer, right? Yeah. Still? Okay. Okay. Make you work. So, serendipity. I'm going to talk two stories. One about cyanobacteria. You tell me, okay, you worked on eukaryotes. Why are you going to talk about cyanobacteria? Why? Well, there was a story that I always found fascinating, and many of the ECODIM, we talk about this developing story about the Eucine A, so this cyanobacteria that were first discovered by clone libraries of the NIF gene, right? By Dave Scanlon, I think, and but nobody knew to which organism these cyanobacteria corresponded to. Then John Zare, at the University of Santa Cruz, decided to use flow cytometry to try to locate within the population of cells of bacteria where these cyanobacteria were. And he was using actually the same flow cytometers that we have in Concepción, in Dichato, the influx. And then he found that one group of cells in his population of cells indeed possessed exactly the same signature, NIF signature, than this new group of uncultured nitrogen-fixing bacteria. So he did sorting, and then he did some metagenomic analysis. And what he found was really quite surprising, is that these cyanobacteria had genes for PS1, but no gene for PS2. He went a little bit further, and actually was able to reconstruct the whole metagenome of this Eucine cyanobacterium. And he found indeed that they cannot fix carbon. They have no Calvin cycles, they have no TCA cycles, they have also no urea cycle. So it was really strange what these bacteria were doing, but still they seems to be extremely interesting. And John came to a meeting in Roscoff that was in 2011, I think, in 2010, oh, I don't remember exactly. No, that was just before 2011, just before the last ECODIM. And we were talking, and he said, okay, we have new data on this, because we found actually that initially we were shaking our sample a lot, and the cyanobacteria were like unicells. But then we decided that to try to process the sample differently. And what we found is that the cyanobacteria signature was associated actually to some very small eukaryotes. So each time he sorted the eukaryotes, he found the Eucine signature, the NIF gene of the Eucine. So it's strange, so probably something either endosymbiotic or... so they decided to sequence the eukaryotes to know what this eukaryotes was. And then, so that's what he did here, and he found that the eukaryotes was, the sequence was extremely close to one sequence we had from the Biosop cruise, one I just mentioned before. It was a haptophyte. very close also to a coccolithophorid called Bradurspora. I will come back to this later. So we were talking and say, oh, but I have a metagenomic sample for this, for this sample, actually. We also, out of the many clone library we did 6 metagenome, and we have this. So that's from this sample called T60, whatever it means. And this is Bradurspora. It's very strange coccolithophorid, very beautiful, actually, a perfect geometric shape. So I said, okay, you have the UCNA genome. I have my metagenome. I'm going to see whether in my metagenome I have also the trace of UCNA. In fact, this sample is from the North Pacific. My sample was from the South Pacific. So I looked into my T60 sample, and bingo, the cyanobacteria was there also. So when I sorted this eukaryote, I also sorted the cyanobacterium with them. And I was able to assemble, actually, almost from my metagenomic sample, this. When I was taking another sample where we didn't have this signature, I could not find the cyanobacterium sample. So these two evidence proves that this eukaryote and this cyano were very closely associated. But the beautiful story, I'm not responsible at all for it, they show that indeed there was a very close association between the eukaryotic cell and the cyanobacteria. And using nanosims, they were able to show that the cyanobacterium, which is fixing nitrogen, is providing nitrogen to the eukaryotes. And conversely, that the eukaryote is providing carbon. Remember, the cyanobacteria cannot fix carbon. So the carbon goes from the eukaryote to the cyanobacterium. So this is the first type of pelagic association between a cyanobacterium, external association, actually between the cyanobacterium and a very small eukaryote. And the eukaryote is tiny. Cyanobacterium is probably 1 micron, and the eukaryote 2.5 micron or something like this. So this was made it in Science. But the astonishing story is that now there is and I didn't extract the data, but if you look in +1, there is a Japanese group that has been able to this is not available in culture. You cannot culture it, but you can maintain it. And the Japanese are very good at doing this. So they did this. They were able to maintain this in culture and to do electron microscopy. And within these cells, they found a cyanobacterium, which was probably, we don't know exactly what happens, but probably we have 2 phases, and Peter, who is here, has worked on the cell cycle, on the life cycle of coccolithophorids, which are very complex. Probably we have this form, which contain endosymbiont, or inside the cyanobacterium. And we have the form that we find here, which is probably a small flagellated form, which contains the cyanobacteria externally. So it's an ever-developing story. So come back to next ECODIM, and there will be another twist to the story. So that's one. And the second one is viruses for Matt. And actually, the person who did this work, Simon Roux, is now doing a postdoc in Matt's lab. There has been this report, so I don't know anything. I'm just reading basically from the paper, which I didn't understand, so you can cut me if I'm wrong. There has been some report of this hybrid RNA-DNA viruses, basically which contain 2 genes, if I understand correctly. One which is related to RNA virus, and one which is related to DNA viruses. And this was done by metagenomics, basically. There is no virus which has been isolated, right, from this type of virus. I think this was metagenomes, yeah. And so Simon was looking into all kind of metagenomic set to look for this hybrid virus, which seems very interesting. And he found indeed that in 2 of our sample, sorted samples, the one where we found Batycoccus, he found these viruses, actually, and he found them here and here. So he found a total of 30 new genome of these hybrid viruses in searching in the data. Great thing with metagenomics, if you have ideas, there are so many data around that you can just dig them and dig them and dig them, and you will find things. So he found them. And what was really of high interest to me is that we had these two samples, again, which are completely processed separately, and he found exactly the same signature in each of these samples, actually. And this was published in very recently in Nature Communication. So it could be that this hybrid virus could be associated with Bathycoccus, actually. That's one hypothesis. And if I don't know who is the group who is doing virus, so maybe we will find one of these viruses in your samples. Anyway, so I will conclude here. So cell sorting is a very powerful way of obtaining metagenomics data for eukaryotes. I don't think you have to do only single cell sorting. I think population sorting is also of interest and provide a lot of information, and it's lots easier to do than single cell sorting. It's much less expensive. If you have reference available, then this is great. You can compare your metagenomes to known genomes, and then you can do a lot of inference from this. This was the case with Bathycoccus, for example. If you don't have reference, it's much harder. You don't know what to say most of the time. But if you have if you have transcriptome or genomes that are now coming out, these eukaryotes genomes are making their coming out. Or if you have chance and a little bit, if you talk at the right person at the right time, then it can also help to interpret this data. I think we need to, in terms of perspective, to really do a much more detailed analysis of the data we have. We need to have more of these sorted genomes, and in particular, we have some sample where this uncultivated clade 9 we're really interested in, and Adriana within my lab is going to soon send them for sequencing at Genoscope. And finally, I think this is really the very, very critical. I think we cannot do any work if we don't isolate the organism. And this is true, for example, even you see for this Leucine A work, because it turns out that having the cells, even not in culture, but maintaining the host in culture is very critical to understand what's going on. So I would like to thank all the people from Roscoff, the people from Banyuls, with whom we have been collaborating very, very actively, Genoscope for the sequencing, also Osvaldo and Rodrigo here, and also my Japanese colleague, which got me started a few years ago on this whole genome amplification and single cells, and the different founding agency, and especially, although I'm not financed by the Moore, but I think their initiatives, community initiatives, are really great. Thank you very much.
You mentioned the need for databases for mapping metagenomes to taxa, but what fraction of your use map to functional categories, or COGS, or what fraction of your metagenomes map to anything functional, COGS? Is that also a big issue for you?
Well, actually, it's, I mean, those that, that's a good question, but those that have a match basically in GenBank, we can more or less map them to COG, but those that don't have match, and we have no it. So, but yeah, we can, I don't, yeah, that's a good point. I have the data, but I don't remember. But it's the same order that the one that for which we have blasted. One of the problems in our sample is that we have also always a mixture of bacteria. We have some bacteria. Some are maybe because in the same drop we sort one bacteria. Some, I think, are because also a lot of these cells are probably mixotrophs, especially the aptophytes, and maybe the chrysophytes are mixotrophs. So we have probably also the bacteria. So that's one of the problems.
And then I wanted to just quickly comment on the genome database. So Karen Holmfeld worked out a nice method for when your viruses are totally absent from the database, you can still sort of make inferences. So I should talk to you about some strategies to do that, because it seems like it would be really relevant in the Daphne-Coccus example you gave. Since you had Mectromonas and Osteococcus, there's ways to sort of make inferences based on the percent identity of groups of the concept.
Okay.
So on your ECODIM particles that you mentioned were haplotypes that you're finding in prolific cycles. And at least Defire, Capsa, and Emiliana have very large genomes. Do your pico haplotypes have smaller genomes, or can you comment on that yet?
That's a good question. I don't know actually because we don't have from the sorted samples we didn't select haptophyte actually because we had to choose, so we don't have a lot of money, so we had to choose. And I don't know, even for the chrysophyte I don't have a good idea of their size yet. So it's, and also the next sample we are going to do will go for Illumina, but this one are 454, so coverage is not so great. But my feeling is that it's more, I mean, except for EOX, I think it's some kind of exception. But my feeling is that the genome is most of the time proportional to cell size. No? What do you think? Yeah. So I think they will probably be in the same order. Because, for example, we have another genome is Amobophrya, which is dinoflagellate related, but we estimated, log EU estimated the genome to 40 megabase pair. So although it's dinoflagellate related, it should be huge, but no, it's in the range of its size.
I'm wondering about one is related to the mixotrophic question of whether there's a genomic signature, like you're reporting about the signal transduction, maybe related, if there's, especially because it's really interesting in these phytoplanktons, many of them are from groups that are normally mixotrophic, whether they are or not. And so that's one question. The other question is also related to sort of genome size issues and changes in that is that with the current methods of, how confident can you be at this point? Is it possible to with confidence assign, for example, say, it's losing these genes, and here's some that we're gaining, or then also maybe you can see changes in symphony. Because, is it possible with paired reads yet to do that kind of thing, or is that out of the way?
So for the first question with the the heterotrophy, I don't know. It was really surprising to me that there was some coherent pattern actually that we'll see in this gene signature, gene category signature. So I don't know, and we need to correct, because these data are kind of uncorrected. You have to correct maybe for the level of expression, things like this. So it's still exploratory. For the second question was
how possible it is to detect, for example, changes in symphony or breaking up the genome, losing genes or gaining new ones.
Well, I think we are still far from there. I think we can get hints, I will say. But not so. But this is the same. I mean, I've been reviewing a lot of this paper. I mean, I've had the chance to review 2 of these papers, which were done with single cell. So where it's better, supposedly it will be better. But with single cell, for example, the coverage is so bad. I mean, it's so patchy that it's very hard to tell from the data whether things have been gained or lost. And so I think we are still a bit far. I think we need to have many more of these to reach this. But I think we will in the end.
A cyanobacterium that fixes nitrogen without having the cathodic tool is an excellent thing. It protects the nitrogen because it does not get inhibited by oxygen. But can it still do photosynthesis with only one photosystem? because it needs a lot of energy. Reducing it with safety to make that 16 negative. That cyanobacteria.
Yeah, that's a very good question, but I don't know. No. Maybe, I mean, now that we know, but it seems to be, first it's not so abundant, so it's not something that you, and I think since it's in symbiosis, it's not going to be so easy, but maybe if we can get the Brachiosphaera, which is the coccolith form, which harbors the cyanobacteria inside, maybe we can get the cycle or something and then get the cyanobacterium by itself. I think if we can get this, but this we have to wait for the Japanese to do it because, or Jan Probert, either one, or Peter.
So it goes Walden when we go to Easter Island or okay, take the green then.
About the Baticopus, you found the different ecotypes. If you consider just the 18S gene sequence, they differentiate different clades?
No, the 18S, the ITS, and the large subunit are 100% identical.
Okay, so it's tricky.
Yeah. So it's only at the gene level, some genes where you see some SNPs, basically. Carlos.
Will be representative when you make some gate and sort these populations? How representative is your sorting for this total community? Because you are sorting like 10,000 cells, 1 million cells, and then you try to lyse them, or you are making direct sequence. I don't know how this works, but you make something to check if you are, when you are making community studies, it's really representative of the inhabiting the water column.
Well, that's a good question. I think what in our case, one thing which I really like actually, but that was chance, is that we had 2 samples, and we found basically almost the same thing in the 2 samples. So now when we go out of the ocean, usually we don't do replicates, but we could, now I think with next generation sequencing, we could do a lot of, metabarcodes on many different, so I think now we can do this kind of study. I think it's more or less representative because what we find, then it corresponds really, this doesn't differ too much of the image, the general image. But now on a given sample, yes, maybe we are not representative.